Hello student, today in this video we will discuss about the phagocytosis. Phagocytosis is the process of engulfment and destruction of solid particles such as bacteria, dead tissue and foreign particles by the cells. The cells that perform phagocytosis are known as phagocytes. The main type of phagocytes of immune system are monocytes, macrophages, neutrophils and dendritic cells. Monocytes do functions in blood similar to macrophages in tissue. Macrophages are monocytes that live in tissues and organs, can do phagocytosis or secrete toxic substances for killing. Neutrophils can do phagocytosis and secrete substances involved in inflammation. Dendritic cells do phagocytosis in tissue and organs, are antigen presenting cells. Dendritic cells basically are a special group of phagocytic cells since besides performing the processing of engulfed pathogen, they well present the same antigen which is a part of adaptive immune system. Function of these phagocytes is to destroy the engulfed pathogen. These phagocytes are equipped with certain intracellular compartments such as lysosomes, which contain microbial substances like lysozymes, nucleases, protease, lipase, H2O2 and etc. There are 7 steps followed in phagocytosis process. Number 1, margination. Number 2, diapedesis. Number 3, chemotaxis. Number 4, opsonization. Number 5, engulfment stage. Number 6, degranulation stage. And number 7, degradation stage. Now in the first stage which is called margination. In the area of infection, the neutrophils get marginated, that is get attached towards the capillary endothelium and start rolling along its surface. This process is called margination or pavementing. The margination is caused by binding of selectins, which are the cell's adhesion molecules present on the endothelial cells with the carbohydrate molecules present on the surface of neutrophils. Second step is immigration and diapedesis. The marginated neutrophils are immigrated in large number from the blood to the site of infection. The process by which neutrophils pass through the capillary endothelial cells to reach the invaders in the tissue is called diapedesis. Third step is chemotaxis. Chemotaxis refers to the process by which the neutrophils are attracted towards bacteria at the site of inflammation. The chemical substances are released from the site of inflammation or infection by the infecting organisms or inflammatory cells. These chemical factors attract neutrophils to the site of infection and they are called chemotaxins. Leukotrienes and components of complement systems and cytokines are some of the chemotaxins. Fourth step is opsonization. Opsonization refers to the process of coating of bacteria by the opsonins. The opsonins coated bacteria gets attached to the surface of phagocyte through the opsonin receptors. The principal opsonins are IgG opsonin and C3b opsonin. The attachment of membrane of phagocyte to the membrane of microbe is called adherence. Now the fifth step is engulfment or ingestion. The neutrophils project pseudopodia in all directions around the opsonized particle, which is bound to the surface of neutrophils. Pseudopodia meet each other on opposite side and fuse. This creates an enclosed chamber with the engulfed material. It breaks away from the membrane, forming a phagocyte vesicle. Then the lysosome of the cell fuse with the phagocytic vesicle to form phagolysosome or phagosome. Sixth step is the degranulation stage. Once the bacteria is engulfed, the lysosome release their enzymes into the vesicle and also in interstitial space. This process is called degranulation. And the last step is killing or degradation stage. Several minutes after phagolysosome formation, the first detectable effect on the microorganism is the loss of the ability to reproduce. Inhibition of macromolecular synthesis occurs sometime later and many pathogenic and non-pathogenic bacteria are dead 10 to 30 minutes after ingestion. The killing mechanisms that phagocytes use can be organized into two broad groups, that is, oxygen dependent and oxygen independent mechanism. In oxygen independent killing mechanism, activation of macrophages synthesize the following, like lysosomes, tumor necrosis factor alpha, and other hydrolytic enzymes. And in oxygen dependent killing mechanism, activated phagocytes produce a number of reactive oxygen intermediates and reactive nitrogen intermediates. When exposed to certain stimuli, phagocytes, eosinophils, and macrophages or neutrophils, oxygen uptake increases greatly, sometimes more than 50-fold, which undergoes a series of changes called respiratory burst. Respiratory burst occurs during activation of macrophages during phagocytosis, abrupt rise in oxygen consumption, increased glucose consumption, large amount of ROI, activation of NADPH oxidase or phagocyte oxidase.